Hello there, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the Inland Pacific Northwest of the most beautiful USA. Today is the 29th of July and the year is 2020. So what I want to do today, and the reason we're going with a video, is we've been doing a whole series of audio lectures on aging. We're taking you some of the very basics of biogerontology, dealing with what we think might be associated with aging at a very initial level, talking about reactive oxygen, talking about longevity, and trying to associate that basic framework with intermediary metabolism, because obviously that links up to nutrition and exercise, and there has been a great deal of research on both of those intake systems for the rate of aging, particularly in our animal model systems, mouse and rat, but also in studies with humans. So I want to do a video lecture today because it's a better format for what I'm going to do rather than with audio, because I want you to be able to see some of the information rather than just hear me talk about it. So let's just get started with this lecture. There it is. So what I want to do today in Authentic Biochemistry is get into T cells and Prenal lipogenesis. Now, we've done this before when we were talking about activation of T cells and basic lipid metabolism in T cells, and also in the system that the T cells is being associated with, because remember, that's an immune response. T cells, after all, are lymphocytes. And we're going to be doing that later, talking about the assimilation and the utilization of T cells as it is linked to Prenal lipid metabolism. But what I want to do at the beginning is lay the groundwork for how this feeds into the aging process. And so it probably won't happen in the very first video lecture, but in the second or third video lecture, I'm going to be bringing in what I'm doing in the audio. So there's got to be two streams of lectures coming to you starting in, now and all through August probably. So let's just get started. So again, I'm Dr. Dan Guerra. This is my lecture series on video for Authentic Biochemistry. And as I said, the date and all of that. All right, so let's just get started here. This paper came out in the Journal of Biological Chemistry just a few months ago. And you can see the citation here. What it's talking about is prenylation. This is a specific kind of covalent modification of proteins post-translationally. So you can get a farnesyl group, which is a 15 carbon, or a geranyl group, which is 20 carbon, Prenal lipid, and that's going to be added to a C-terminal cysteine residue of a CAAX or CAAX-like motif, depending on the characteristics of the X, where X is, of course, is any other amino acid. The C is the cysteine that's going to get prenylated, and then the A, the two A amino acids are aliphatic amino acids. They're going to be between the cysteine and the X, which can be any amino acid. When you prenylate a protein, it allows them to be anchored to the cellular membrane. Okay? So geranyl diphosphate synthase is the enzyme which is going to come from the mevalonic acid pathway. That's de novo cholesterol-genesis now we're talking about. And it's responsible for synthesizing that C20 Prenal lipid. And from its substrate, farnesyl pyrophosphate, so you've got a 15 carbon preformed Prenal lipid, and then you're going to add 5 carbons to it, because remember you're adding isopentanoate pyrophosphate, a 5 carbon intermediate, when you build up on Prenal lipid metabolism. Up to C20 is usually where it stops, with C25. Okay, so obviously the enzyme that's going to be synthesizing that geranyl pyrophosphate is going to be leading away from the farnesyl. So it's going to be a utilization either of the farnesyl group or the GG group. to do this covalent modification. And that's going to make a big difference on the downstream processing and eventual function of the protein that became prenylated. All right? So this, of course, is, again, it's all post-translational modification of proteins. So I want you to remember way back, this would have been in the winter, when we were talking about non-alcoholic fatty liver disease. And it's either non-alcoholic fatty liver, which is simply steatosis, that is a buildup of lipid in the liver that's not associated with alcohol abuse, or it's NASH, and that is steatohepatitis. Okay, so again, non-alcoholic steatohepatitis. Now, take a look at this diagram here, and you'll see why all this is kind of significant. Prolonged lipid accumulation and inflammation that becomes associated with it when you get to hepatitis, right, is going to advance, or may advance, usually does, in severity to liver fibrosis, and then cirrhosis, and ultimately to hepatocellular carcinoma, which is a very important cancer in humans. So pathobiology works like this. You get chronic liver disease, which can lead directly to cirrhosis, and it can go to the cancer. You can also get a viral hepatitis B, C, either one of those two viral hepatitis species. You can get hemochromatosis, alcohol. Now, alcohol then would make it not associated with the NAFLD, but it still can lead to cirrhosis ultimately, and of course to hepatocellular carcinoma. And you also get various other kinds of prequels to the major disease. So these can lead to regenerative stimuli, which can ultimately take cirrhosis to the hepatocellular carcinoma. You can also get environmental co-carcinogens, which is going to lead to a non-cirrhotic pathway, yet making it to cancer. Remember, cirrhotic is going to be one that's going to deal with tissue degeneration, right? Genetics plays a role both in the progression of cirrhosis to the carcinoma, and also plays a role in the non-cirrhotic pathway. So there are multiple interactions here that occur in the diseased liver. So as the non-alcoholic fatty liver disease develops, insulin resistance also occurs, and it occurs because of the phosphatidylinositol 3 kinase AKT pathway, which is of course activated in the liver. So this diagram here is showing you that insulin resistance is one of the first features of a diseased liver. Next, you're going to get this LXR, which is a receptor that's going to induce de novo lipogenesis. And you're going to get AMP kinase, which is going to also be involved in glucose uptake. So these are all kinds of pathways that are going to be involved with modifications of proteins that have been prenylated. And we say it's by MBA, that's the mevalonic acid pathway. Okay? So you've got insulin resistance because of this triggering mechanism here, this kinase pathway. Then you've got LXR alpha and AMP kinase, which promote de novo lipogenesis and glucose uptake, respectively. Then you get an activation of RAC1 and and RAS, and that's FAS-L, that's FAS ligand, okay? And so you're going to get cirrhosis, and I told you cirrhosis is the damage of the liver tissue. And one of the leading mechanisms for that is an apoptotic, right, a degeneration of programmed cell death. Ultimately, you're going to get an uncontrolled proliferation after this partial degeneration, and that's going to involve the RAS-ERK1-2 pathway, and that's going to give you full-blown, hepatocellular carcinoma down here. So you go from a normal liver to the non-alcoholic fatty liver, and you're going to go to NASH, remember that's the steatohepatitis, and then you're going to get to the cancer. So I want you to understand that there was a progression like that. Now, there was a paper published, we're looking at a 2020 paper here where this comes from. Paper published a few years before that. I just want to show you the title of the paper. Kind of that's all I want to show you at this stage of the lecture. Hepatic steatosis risk is driven by an increased de novo lipogenesis following carbohydrate consumption. So you probably heard a lot about dietary changes over the last decade. The American Nutrition Association and the American Medical Association had for many years been suggesting that lower lipid in the diet is better for health. And they thought that because lipids were involved in atherosclerosis, and we know that lipids are also sometimes and often in a very indirect way associated with cancer, and this is an indirect way with melvillamic acid, obviously. I talk about covalent modification of proteins that when they are modified with prenolipids, all these proteins you're looking at here in this progression from normal to HCC, some of those proteins are going to be prenolated. And whether or not they are farnesylated, that's a C15 prenolipid, or geranylated, is going to trigger either a non-oncogenic or an oncogenic pathway. That's why we talk about MVA being really important here in this progression of this liver disease. So let me get back to this whole thing about nutrition and the Medical Association recommendations. So they've been recommending for a long time, even though us lipid biochemists did not agree with it, that lowering lipid in the diet was going to help ameliorate some of the major causes of disease, morbidity, and death in humans. That is obesity, cardiovascular disease, and also cancer. Point that paper in 2018 suggests is that when you get glucose will be converted to lipid, and that process of conversion of carbohydrate to lipid, which occurs in the liver, will allow for fatty liver deposits, and that can lead directly to the steatosis, steatohepatitis, cirrhosis, fibrosis associated with that sometimes, and then the prodromal phase of hepatocellular carcinoma. And it's because you're starting from carbohydrate that you get this accumulation of lipid. Lipid in the diet is transported and mobilized in a totally different way, because there you're talking about chylomicron, which are lipoproteins, mediated movement of lipids before it ever gets to the liver. This is all happening in the gut, particularly in the small intestine. And even when lipids do make it to the liver, they are esterified to cholesterol, that is, fatty acids are esterified to cholesterol, and lipids like triacylglycerol and phospholipids, sphingolipids, are loaded onto different set of lipoproteins, the very low density and the low density lipoproteins in particular, and those are transported out of the liver. They have different receptors, and they move through the body at different receptors in different tissue beds, right? So exogenous lipid coming in the diet is treated differently by the body than lipogenesis, which is occurring hepatically. And it is that lipogenesis occurring hepatically from glucose that is more responsible for the obesity epidemic we have, and not so much for lipid intake. Because when you take in lipids, you decrease de novo lipogenesis in the liver, and that means you have a different total trafficking of the lipoproteins to the periphery, and therefore different adipose deposition and different lipid deposition in the cardiovascular system, for example, pulmonary tract, etc. So that's an important take-home message. I wanted to get that to you at the beginning. Go to the next slide. All right, back to this paper now in JBC 2020. So this is just showing you the pathway for covalent modification of proteins. So on the left, you see this farnesylation. So you have a farnesyl transferase. You have farnesyl pyrophosphate. It's loading onto this cysteine residue. It's going to be a sulfur atom, right? Because cysteine. It's going to have that motif on the protein, that CAAX I told you about. And a protein that gets farnesylated is GTPase. Okay? So GTPase can also be geranylated, right? And so it's going to be prenolated, and it's going to show up in the membrane where both of these GTPases have to be functional. That's where they have to be in terms of localization in the cell, in the plasma membrane. However, because of the difference in prenylation, the protein will function discretely differently, okay? And that's shown over here. So, well, first, before I get to over here, I want to tell you that you're going to be thinking about, I want you to understand and consider that just because you have a prenylated protein and it gets inserted in the membrane, protein like this GTPase we're looking at here, generic GTPase, you might think that, well, okay, it was meant that farnesylation or that germination was placed there so that hydrophobic lipid moiety, that prenolipid, targets the protein to the membrane. I told you that's where the protein has to function, is in that membrane, and we're done. And what I'm telling you is that's not the case. The difference in the prenylation, being C15 or C20, makes a big difference in the function of that same enzyme, the GTPase. And it's going to alter metabolism because of signaling in that plasma membrane between the cell that has a farnesylated GTPase versus a cell that has a geranylated GTPase. Okay? That's a key feature. Now, on the right here, you get also a non-covalent protein ligand binding, where you get geranyl pyrophosphate, or you get farnesyl pyrophosphate localized to these three different proteins. So you can get a GGPP, you can get a geranyl for the skip protein. What's skip2? The skip2 is the substrate recognition component of an SCF complex, which is the skip1 CoL1F protein, E3 ubiquitin protein ligase. So this is going to be involved in protein degradation. Okay? So it's a really important protein. And it needs to have this non-covalent geranyl for it to be activated appropriately. Okay? And what it does, that protein, when it has that GG group, non-covalent association here, not like on the left, it mediates the ubiquitination and subsequent proteasomal degradation of target proteins involved in cell cycle progression. And it's involved in the regulation, ultimately, that particular modification of that particular skip protein is going to be involved in moving the cell cycle from a G1 phase to a synthesis S phase, a synthetic phase, a DNA synthesis phase. Likewise, if you have a protein farnesylated, the FXR protein, that protein is named from the Fragile X retardation syndrome protein. It was first described in humans. And what that protein is a global messenger RNA untranslated region binding. It's the 3' end of messenger RNAs. Now that's going to have something to do with mRNA stability. And when you alter mRNA stability, you alter its translatability. Okay? Remember, messenger RNA gets reused every time you synthesize polypeptide in the cytosolic or in the ER-localized synthesis of proteins, right? The translational machinery. So if you're going to be using FXR, right, and it gets farnesylated, it's going to be fully functional, and that farnesylation is only can occur if you have sufficient amount of farnesyl pyrophosphate. But that level of that lipid can be diminished if you have a hyperactive geranyl phosphate synthase. That's what that enzyme is leading up there. So if you make more of that, you're going to be non-covalently, right, triggering skip2. And also, what's this one here? This is PPAR gamma. That's peroxisome proliferative activator receptor. Now, that particular transcription factor, that's a transcription factor, is going to be necessary to trigger enzymes involved in changes in bioenergetics, particularly the utilization of fatty acids. And what I mean by that is beta oxidation of fatty acids in the mitochondria, and that's going to lead to a totally different mode for the cell, right? So you can see that protein gets non-covalently linked to geranyl group, not to the farnesyl group. So you got a ubiquitination process that's going to be enhanced by geranyl, and you're also going to have a transcription of proteins are going to be associated with the utilization of fatty acids. That means the biofuel will be changing from glucose to fatty acid. And that means molecular oxygen is going to be necessary. And with a lot of molecular oxygen, a lot of NADH production because of the beta oxidation of the fatty acids. Also, of course, the flavonoid dinucleotide production, the reduction of that, that's going to lead to the potential for a lot of reactive oxygen being produced in that cell. So you see how these are all linked together. You have to understand the multiple levels of intermediary metabolism from transcription all the way to signaling. Okay, and those are like the two boundaries there we were talking about. And intermediary metabolism is triggered in multiple layers, so that for here we're just talking about bioenergetics, what fuel you're going to use. But ultimately, it's also going to be involved in all the other systems. Amino acid metabolism, transamination, the TCA cycle, phospholipid synthesis, sphingolipid synthesis, and of course, DNA repair, DNA recombination, and DNA replication. It about covers all the room. Plus, as I said, protein synthesis, because of the stability of the messenger RNA, and protein degradation, because that skip protein. You see how that all functions just because of prenylation of certain proteins and their functionality in given locations in the cell. All right. Now, take a look at this paper here. Again, take a look at this diagram. Again, it's from the JBC paper in 2020. Okay? So you go from a healthy liver system to a non-alcoholic fatty liver disease to a fibrotic. Once you get to the fibrotic, you can go down a cirrhotic pathway, right? Or actually at the same time it becomes fibrotic, and then from that directly to hepatocellular carcinoma. Or you don't have to go through the cirrhotic pathway, you can go through fibrotic directly to HCC. Now look what's going on here. I put on my glasses, which I didn't clean, but that's okay. As you increase the amount of geranyl pyrophosphate synthase, you got here if you look at a balance, but when you increase the amount of that enzyme, you're going to have more geranyl pyrophosphate. Okay, more of that prenal lipid is going to make it so that if you're looking at this teeter-totter effect, right? You could also look at it as like a rheostat, right? Down and up. You increase the amount of that enzyme, you're going to make more of that lipid from this is the substrate, right? So that means it's going to be lighter, right? In this diagram, you have more of that. That's going to change the ratio of farnesylated proteins to geranylated proteins. In fact, it's going to lower the ratio because you're going to decrease the numerator and increase the denominator. That ratio then will drop, right? What that means in this system is you're going to have about an equal use of glucose or lipid, right? With, again, showing the rheostat control, the fulcrum of that is that geranyl pyrophosphate, right? Synthase, the enzyme. Now, as time goes on from the non-alcoholic fatty liver disease to getting fibrosis, leading, as I said, to hepatocellular carcinoma, right? The opposite occurs. You're going to get a decrease in that enzyme because you get a decrease in the conversion of farnesyl pyrophosphate to geranyl pyrophosphate. Remember, that's the C15 to the C20 prenal lipid synthesis pathway. Because of the decrease in that enzyme, right? Now what you're going to have is an increase in the ratio. You're going to have more farnesylated proteins and less GG-elated proteins, geranylated proteins. That is going to greatly alter the metabolic pathway. And what it's going to do is it's going to increase, it's going to alter the glucose to lipid utilization system, okay? Now, that's going to allow you to get the Warburg effect. Now, remember when we were talking in the Authentic Biochemistry Podcast Aging series, I spent one of those 5 or 6 lectures on what? Comparing aging or senescence to, that's right, oncogenesis. And I told you about the Warburg effect, and that is glycolysis even in the presence of molecular oxygen, right? So you're using glucose, you're making a lot of lactic acid, remember, but you're not converting pyruvate to either oxaloacetic acid or acetyl-CoA, right? Because remember those mutations we talked about, the succinate dehydrogenase, right? And the fumarase, and we talked about alterations in the PK, that is the pyruvate kinase, right? You get a lot of lactate buildup. I know that you think back about those lectures I was giving you in the audio feed. Then that's going to give you the Warburg effect, where you're going to have a lot more glucose utilization, right, via the glycolytic pathway, and not so much lipid. Once again, tipping the balance. See? All right. So it's a major effect here of altering the biofuel, and then also all those proteins we talked about, proteins involved in overall protein degradation via the proteasomal pathway, overall and specific protein synthesis at the stabilization of messenger RNA. Stabilized messenger RNA is going to give you more presumable product, which is the translatable product, which is polypeptide. And those on a global level and on a specific level as well, because remember, the transcription of specific genes is going to be involved in some other signal cascade pathway. All those other proteins we were talking about 2 slides ago, right? They're going to be triggering because they are transcription factors. Unique set of genes that are going to be triggered in this, leading from the non-alcoholic fatty liver disease to the fibrotic or cirrhotic liver, all the way to the hepatocellular carcinoma. Okay. Now, this comes from a paper published in 2010, but I want to give you an idea about what's going on, again, in terms of metabolism. Okay, so a free fatty acid receptor-mediated insulin secretion enhancement is what we're looking at here. Okay, take a look at how this works. In the intestinal cell, don't worry about glycolic acid right now. There's a membrane protein called GPR120. We've talked about this in lectures several times ago. I remember it, maybe you don't. And this is a receptor that's going to utilize free fatty acid. That's going to trigger in the intestinal L cell, okay, the secretion of GLP. Okay, it's a glucagon-like peptide 1. Glucagon-like peptide 1 is going to be secreted from the intestinal cell, right? And it's going to make it to an islet cell of the pancreas. This is about insulin secretion, remember. And that's where you get beta cell of the pancreas. GLP is going to bind to the GLP receptor, right? And that's going to have, oops, sorry. Gosh, I hate that when it goes in. Nope. Whoops, way back. Okay, and what that protein is going to do when it binds, when the glucagon-like peptide binds to its receptor, it's going to trigger the release of calcium, which is going to alter ATP to ADP ratios, right? And that's going to be altering glycolysis and respiration in the pancreas, right? All of that will lead to insulin secretion, right? So free fatty acid is going to allow for insulin secretion, and glucose is going to aid in that response as well. And plus, one more element here, directly at the beta cell in the pancreas. Free fatty acid can also bind to another receptor here. This is the GPR120, it's the GPR40, and it's also going to respond the same way ultimately. It's going to increase the amount of calcium, it's going to alter the ratio of ATP to ADP, and it's going to induce insulin secretion. So free fatty acid and glucose, if you want to understand how those two metabolites increase insulin secretion, so think about after a meal, this is how it's done, via the interaction with the intestine, generating GLP-1, right? Just showed you there. And then right at the beta cell of the pancreas, that free fatty acid and that glucose are going to work cooperatively, but through different receptor systems. The glucose is going to come through the GLUT2 pathway, as it's shown there, to ultimately trigger calcium release and then insulin secretion. Okay, so that's what I want to tell you. TUG424 was identified as a GPR40 agonist, so there's an agonist for that. And GPR and free fatty acid binding activity activates GPR120 in intestinal L cells, stimulate glucagon-like 1 peptide. I told you all this, okay. The GLP-1R and islet beta cells increases calcium. The G protein signaling Okay, the other thing I was going to say about glycolic acid derivatives are determined at GPR 120 partial agonists. Okay. So that's where you get these two. Right? Okay. Next slide. All right. Now, remember a few minutes ago, we told you that one of the genes that is associated with this MVA transduction cascade, because remember the farnesylation or the germination of proteins is going to modify the activity of the AMP kinase, right? Remember the AMP kinase is a really important signaling motif to alert the cell to go into an alteration of what utilization of biofuel is going to be induced. So what AMP kinase does when it's triggered is it's going to cause a lot of things in the cell, and only a few of these are shown here. First of all, you're going to get a blockage of the acetyl-CoA carboxylase. Now remember what that enzyme was, right? That's the enzyme that's going to be involved in the synthesis of malonyl-CoA. This is the first-rate limiting reaction leading to mal-CoA. The 3-carbon compound is the one necessary to trigger fatty acid synthesis. It's not shown here that once you make mal-CoA, a series of reactions later, because fatty acid synthase is a cytosolic polyprotein complex, a heterodimeric polyprotein complex in mammalian cells. It's going to synthesize free fatty acid. That's going to be shut down. At the same time, the malonyl-CoA decarboxylase is going to be turned on. Remember this from several lectures ago? We're talking about this in authentic biochemistry. So that means you're going to make more acetyl-CoA. The acetyl-CoA is going to build up. Notice that pyruvate also, right, can be converted to acetyl-CoA. So watch what happens with this whole process. AMP kinase, the AMP kinase is going to inhibit fatty acid synthesis. It's going to inhibit this PEP carboxykinase enzyme. PEP carboxykinase takes pyruvate, converts it to PEP, and then in gluconeogenesis to make glucose 6-phosphate, and then either glucose or glycogen, right? Right. But AMP kinase is going to block the glucose 6-phosphatase, so you're not going to make free glucose. So this is a liver cell, right? You're going to be blocking gluconeogenesis because this enzyme is blocked. That's a gluconeogenic enzyme. You're going to be blocking fatty acid synthesis. And what you're going to be doing is enhancing glucose uptake, right? AMP kinase causes glucose uptake. At the same time, it's going to enhance beta oxidation of the fatty acids because you need that as well to be able to make the whole cellular system continue to function. So the AMP kinase acts as a central energy sensor, as it writes here. Activated AMP kinase deactivates gluconeogenic PEP carboxykinase, the glucose 6-phosphatase, thereby decreasing hepatic glucose production. So you're not getting the liver producing glucose. It increases glucose uptake in the liver, though, by inducing glucose transporters, both the GLUT4 and the GLUT1. As this says, AMP kinase also stimulates lipid metabolism by decreasing mal-CoA levels through inhibiting the carboxylase and the activation of the decarboxylase. I just went through that with you. There's a compound called berberine or BBR, and it exerts a beneficial effect on diabetics through activating this AMP kinase. And you can see why, because it's going to diminish the amount of serum level glucose, right? It's going to get rid of that hyperglucosemia. It generates glucose through glycogen breakdown. Okay, so glycogen phosphorylase, the key enzyme for glycolysis, is another key enzyme for glycolysis because it gives you glucose, right? It generates glucose through glycogen breakdown. Mazolinic acid, bredamolic acid, and the related derivatives inhibit that phosphorylase enzyme, mimicking the action of insulin by stimulating hepatic glycogen synthesis. Okay? So you get an idea what's going on here. You're not allowing for more glucose to get into the serum. So AMP kinase are really important bioregulator of this whole system. And what I'm bringing this together for you is I want you to understand that mevalonic acid, right, that whole pathway which is involved in the synthesis of intermediates on the way to make cholesterol, because that's what the mevalonic acid pathway ultimately does, de novo cholesterol genesis, is going to make these prenolipids, farnesyl groups and geranyl groups. And those, yeah, sure, they are frank intermediates in the synthesis of steroids, right? Not just cholesterol, but also they are involved in either covalent or non-covalent modification of proteins, which is going to trigger cellular phenomenon that's going to feed back into the system, such as the AMP kinase triggering, right? Because you're going to be getting that farnesylation of the GTPase, and you're going to get that whole induction of AMP kinase. You see how the metabolic pathway, lipid metabolism, you're not talking about synthesizing cholesterol here, right? You're not at all. You're not going to be making cholesterol. You're going to, however, be triggering the synthesis of C15, C20 isoprenolipids, but they're not going to go any farther than that. And those are not going to be used as building blocks to make steroids or sterol like cholesterol, but they're going to be used as covalent modifying agents or non-covalent modifying agents to trigger responses such as protein degradation, transcription rates, and whatnot, a specific sequence of genes that are going to allow for normal metabolism. All right. Now, where I'm going to go next is I'm going to give you a review of T lymphocyte regulation by mevalonic acid metabolism. So there, where I was showing you about insulin secretion, about hepatocellular carcinoma, right? And we haven't even started talking about the immune system. So while that's going on in pancreas and in liver, right, and the intestine, right, all three of those systems have already been invoked in this lecture. Now we're going to switch to an understanding of mevalonic acid in lymphocytes, okay? So what's occurring in the lymphocytes and the triggering mechanisms, right, that will turn on or regulate at any rate the mevalonic acid pathway may not be available in other tissues. So when you're thinking about modifying lipid metabolism to modify, say, the activation of T lymphocytes, because you want them to be activated to trigger a oncolytic event, right? So T lymphocytes show up in tumors and destroy tumor cells. Controlling the mevalonic acid pathway in T lymphocytes is not going to be the same, even though the compounds you use may be agonists or partial agonists or antagonists in certain parts of that pathway. You see, might not function the same way in what's going on in the organs themselves, right? So not the tumor, but what's happening in the liver in normal physiology or in the pancreas. So this is how you get a pleiotropic effect of having one system functioning one way, another system functioning another way, but looking at the same given nested pathway. In this case, it's mevalonic acid, leading to all those intermediates in the pathway, as well as the production finally of cholesterol, right, and steroid hormones, which are also further going to regulate the system. Because what are some of those steroids, right? The glucocorticoids. Glucocorticoids are going to also control carbohydrate metabolism. That's why they're called that, right? But what else do? They cause immune suppression, right? So you've got multiple layers of mevalonic acid metabolism, and that because there's multiple tissues and multiple cell lineages are going to be using it. And the triggering mechanism, the regulation of those pathways, is going to be specific to those organ beds, right? So I'm going to go into just this introduction here, and then I'm going to stop, and then we'll reorganize our thoughts, and we're going to go into the next lecture, which is going to give you this full appreciation for what's going on in T lymphocytes. I'm just going to introduce it here. Signaling through the T cell receptor. Now remember, the TCR, the T cell receptor, is invoked by a recombination mechanism in those T lymphocytes, right? What that means is the T cell receptor is going to have multiple potential recombination products. And that's going to be involved in how the T cell receptor functions relating to its interaction with the innate immune response, okay, particularly dendritic cells, but anything that's dealing with either MHC1 or MHC2 class exhibition and presentation of antigens to the T cell receptor. That's one component of it. But the same thing you have to understand, the T cell receptor is going to be involved in the activation of what? B cells. And B cells then are going to generate things like antibodies, that is immunoglobulins. So you get an idea that what's going on with the T cells is really fundamental to be in the midst of the innate immune response, say innate immune response triggered by tissue degeneration like in the liver, or maybe a tumorigenesis in that same liver, going from, remember, the cirrhotic fibrotic event to hepatocellular carcinoma, and you want T lymphocytes activated, mobilized to that tumor in that liver. But at the same time, the liver is going through a multifunctional change based again on the integration of mevalonic acid metabolism, right? Let alone what happens with cholesterol buildup and cholesterol ester buildup on lipoproteins that are being secreted by the liver and making it to the periphery, where adipose tissue, cardiovascular tissue, kidney, skeletal muscle, endothelial cells, right? So this is how you have to consider all these multiple integrated events. They're all events, they're all dynamic systems that are involved in graded metabolic pathways, such as the synthesis or turnover of fatty acids or proteins or amino acid metabolism, carbohydrate metabolism, whether or not you've got the Warburg effect going, like in full-blown tumors, or if you've got some tipping of the balance and going to beta oxidation of fatty acids, generating more energy in the periphery of those tumors, right? And at the same time, what's going on to all that activation to generate cytokines and chemokines and growth factors and transcription factors? They're going to be leading to things like the synthesis of more cytokines and chemokines, some of which are pro-inflammatory, and some of which can kill a growing tumor, or can also overshoot and become an autoimmune response, killing organ tissue. Okay. So the metabolic regulation of T lymphocytes is a consequence of TCR stimulation. Of course, it attracts a lot of interest because of what I just told you. It's increasingly appreciated that T cell activation results in metabolic reprogramming and that distinct T cell functions, so Th1 cells, Th2 cells, Th17 cells, Treg cells, natural killer T lymphocytes, all of those, just to name the ones we mostly talk about. There's also innate-like T cells, which we talked about before. Increasingly appreciated T cell activation results in metabolic reprogramming, and that distinct T cell functions depend on the activation of appropriate metabolic pathways, energetics alone. In contrast to resting cells, which pursue efficient accumulation of ATP synthesis, activated T cells must ensure high metabolic flux, anabolic pathways, to acquire a specific molecular repertoire. What is that molecular repertoire? Maybe the production of cytokines or chemokines, growth factors, or transcription factors, leading to an entire interaction of the T cells with the B cells and the innate cells. So not just the production of ATP, but the transformation of the T cell lineage, say going from, say, a Th17 cell to a Treg cell, right? Treg cell member is going to suppress the immune response at the T lymphocyte level, right? All of that has to be work in combination with what the T cell is doing in terms of interacting with other cell types. So that's just my introduction to this whole system. Let me get this. All right. End the show there. So what I'm going to do now is end this lecture. And when I get back with you, we're going to go into T lymphocyte metabolism in great detail. Now, why am I doing this in conjunction with the senescent aging Authentic Biochemistry podcast audio lectures? Because not just because I'm showing you a differentiation between cancer and cell death, the senescent process. I'm going to bring this back in and I'm going to put them together. I'm going to cause a recombination of events. I'm going to use the video lecture to help marry with the audio lecture so that while we're talking about aging, we're also talking about the potential for certain diseases. Some of those diseases while you're aging, as you know, are, for example, in the central nervous system, Alzheimer's disease. But what else can happen is you can get hepatocellular carcinoma, or you can get cardiovascular disease. You can get atherosclerosis, for example. You can also get what? Various forms of cancer, lung cancer, and then the one I ultimately want to land on is glioblastoma, because then we're going to be able to trigger this all back to our discussion of the HPA axis, right? And remember what that was, the whole pituitary adrenal axis, right? Hypothalamic pituitary adrenal axis, and the production of hormones regulating normo-physiology, and when that's functionally altered in a disease state. And then I'm going to develop this around a event ontological discussion, ultimately, of how aging is going to be intimately linked with these disease states, creating essentially a florid environment for cancer to be generated, or atherosclerosis, or various forms of heart disease, or just alterations in metabolism leading to metabolic disease. And now all of that can be, other superimpositions can occur on that by the chronic overeating and obesity epidemic we have. And then how that can all lead to some very specific diseases, such as Alzheimer's on one hand, which is degenerative in the CNS, versus glioblastoma, which of course is proliferative cancer. So that's where I wanted to take you ultimately and get you where we're going to be ultimately. There's going to be several lectures later. So hopefully you enjoyed this. First time we've had a video lecture, first time we got back. So hopefully people are able to get outside and have a good summer. Certainly I get outside a lot. I totally encourage a lot of exercise and a lot of sunshine and a lot of time with your family whenever you can do it, and enjoy the summer, because school will start. And when school starts, our kids will be back in school, and we'll be back doing our other types of activities. And what I want you to do is be well nourished, well exercised, and also increased in knowledge. And that's what we do in Authentic Biochemistry. So again, this is Dr. Dan Guerra, saying from Authentic Biochemistry Studios, in combination with Guerra Med. Hope you have a very pleasant week, and saying bye for